New enzymes are synthesized. These inactivate the drug. Only occurs when new genes are acquired. Some microbes inactivate the drugs by producing enzymes that permanently alter drug structure. Example, bacterial enzymes called beta-lactamases hydrolyze the beta-lactam ring, a critical structure, of penicillins and cephalosporins, rendering the drug inactive. 2. Permeability or uptake of drug into bacterium is decreased, usually occurs via mutation. The resistance of some bacteria can be due to a mechanism that prevents the drug from entering the cell and acting on its target. Antibiotics often have to bind to either an external protein on the surface of the bacterium or to an internal structure like a ribosomal protein. If a mutation has changed the amino acid sequence of the binding site, the antibiotic will no longer be able to attach, and the bacterium will become impervious to the drug. 3. Drug is immediately eliminated, usually occurs via acquisition of new genes. Many bacteria possess multi-drug resistant MDR pumps that actively transport drugs and other chemicals out of the cell. These pumps are proteins encoded by plasmids or chromosomes. They are stationed in the cell membrane and expel molecules by proton motive force similar to ATP synthesis. They confer drug resistance on many gram-positive pathogens, Staphylococcus, Streptococcus, and gram-negative pathogens, Pseudomonas, E. coli. Because they lack selectivity, one type of pump can expel a broad array of antimicrobial drugs, detergents, and other toxic substances. 4. Binding sites for a drug are decreased in number of affinity, can occur via mutation or acquisition of new genes. Microbes can get around protein, RNA, DNA, and membrane structure attacks from antibiotics by altering the nature of the target, such as through changing a binding site. Example, penicillin resistance and Streptococcus A are related to an alteration in the binding proteins in the cell wall. 5. An affected metabolic pathway is shut down or an alternative pathway is used, occurs due to a mutation or original enzyme or enzymes. Example, sulfonamide and trimethoprim resistance develop when microbes deviate from the usual patterns of folic acid synthesis.